It is a pleasure for me to be here and share my research work. This time I want to talk about the lung adenocarcinoma hybrid theranostic nanomaterials. Currently, the research work still lack of the efficacy, diagnostic or prognostic technology for use to the lung cancer cells. So this research work is combined with the targeting and the therapeutic method to treat and track the lung cancer cells. I have divided my research work into 5 parts. First, I'm going to talk about the lung adenocarcinoma, the disease. And second, I will introduce the persistent luminescence nanoparticle as the targeting and the therapeutic nanoplatform. And the third one, we introduce the MAGE-A3 nanomaterials. We use the MAGE-A3 combined with the platform to track the tumor cells. And the fourth, we use the cell model and the mouse as the animal model to prove our research ideas. And the final, we will use the afatinib as the drug to cure this disease. And finally, we will introduce the conclusion to end this research. In the first part, I want to introduce the lung adenocarcinoma. We also call this a type of the lung cancer cell. This is the X-ray photos of a patient who has a lung tumor growth in the left lung. And we can see after tracking for several months, the tumor in the left lung will proliferate to the right lung. And we can see the lung are the most frequent site for the metastasis from the milestone. Image plays an important role in the screening and detect the cancer cells. So next we will try to use our nanoparticle to make the fluorescence image to detect the disease. Our major nanomaterials, persistent luminescence nanoparticle, is based on the synthesis of the mesoporous nanoparticles. We use it to a template and dope the metal ions into the pores. And then we dope the afatinib as the drug and use the MAGE-A3 modifier on the surface to make it have the targeting and the therapeutic ability. This figure shows the persistent luminescence nanoparticle have the crystal size. And then we use the mesoporous nanoparticle as the template, so it has the pores in the middle. After we dope with the metal ions, we can see they have the dark spot in the middle of the pores. So why we called this nanoparticle persistent luminescence nanoparticle? This is because the particle will absorb the UV light and generate near-infrared light for a long time. It is because the energy gap of the nanoparticle is specific. We can see when the team doped into a nanoparticle, the electron trap will make the electron slowly release from the nanoparticles. So that's why it will generate a long-term luminescence for this system. And we can see after we conjugate, we dope the tin ion into a nanoparticle, it will generate the fluorescence until 10 hours. But after we remove the ions, the long-term luminescence cannot detect easily. In order to make our nano platform have the targeting ability, we use the MAGE-A3 aptamer to modify it on the surface. MAGE-A3 aptamer can recognize the MAGE-A3 receptor on the cancer cells. It's because the cancer cell express a high MAGE-A3 receptor. Here we use the convolval image to make sure this result. And we can see the BIS2B is the normal cell, and the A549 and the CL1-5 is the cancer cell. So we can see when a cancer cell treats with our particle, it accumulates more particles in the tumor site and generates more high specific luminescent signals. In order to prove our research work, we use a cell model and the animal model to make the pathology test. First, we use the different groups for treat with the cancer cell, and we can see when the afatinib conjugate with the nanoparticle and the targeting by the MAP, the treatment efficiency is the best for compared with the other groups. And we can see the nucleus will make damage, and the tunnel reagent will standing in the nucleus. For the in vivo animal model, It shows we use the particle to treat the mice. After the 1 hour, it still can see the very strong signal. And even to the 10 hours or more further, we can see the signal still can be detected by the Evincible system. Finally, we use the mice orthotopic lung model to evaluate our therapeutic result. And the first we can see after we inject our particle into a lung, the Evincible system can detect the fluorescent signals. And next, the control group have the very big lung cancer tumors. And after we treatment, the tumors begun and disappear. We use the IV system to detect the tumors change also, and we can see the luminescent signal become smaller and disappear for our treatment. Then we use the H&E tissue staining to evaluate the result. In the control group, we can see the red square shows the first lung adenocarcinoma, and then it will metastasis to the orange square. And after we use our group to treatment with this disease, we can see the lung become normal and no disease obvious to detect it. Even though we have mimicked the human body with the animal model successfully, there are still lots of attacks need to be conquered. It is a different matter if we use the luminescence system in a human body. Yet, given the current development of the programming of the luminescence-assisted diagnosis, still should be around the corner.